Siamo tutti? C'è qualcuno fuori?
Qualcuno sta arrivando.
Allora, aspettiamo un minuto. Non facciamo il quarto d'ora, non facciamo 10 minuti. Ce l'hai? Ok, direi che possiamo iniziare. Thanks from Andrea Luchetti for your presence in this afternoon. The comparative genomics change in genome architecture during animal evaluation.
Okay, thank you very much, guys. Good afternoon. Today I will talk to you about something probably some of you already know, okay? For some others would be completely new. And all it is about genomics and comparative genomics, okay? I try to keep the presentation light, not particularly technical, maybe highlighting only the positive parts of the story, but there are also many bad jokes that we will encounter during the presentation. As a approach to this lesson, I would like you to interrupt me anytime, every time you have a question, every time you want to discuss something. Okay, I'm not used to simply talk and have people listening, but I like when we have some exchange, even during the lesson. Okay, so feel free to interrupt me anytime. Okay, especially if you don't understand something, which is based, I guess. Okay, comparative genomics. Well, first of all, of course, here at my contacts that may be useful if you have any question later. Okay, I'm a zoologist or pretending to be a zoologist. Okay, a department of BGR department, like Stefano Goffredo. Okay, and I'm an evolutionary biologist. Okay, that means that everything I do during my research, okay, is to always have an evolutionary perspective to the problem. Okay, and you may know that genomics not necessarily rely on evolution, but as people more entitled than me already stressed out, nothing makes sense in biology except in the light of evolution. I will not talk about evolution, strictly speaking, okay? So we will see that many of the approach, I mean the technical approach to genomics, okay, are heavily grounded in evolution. Okay. So this is a small summary of what we will talk about this afternoon. Okay, it's a very short and unfortunately incomplete presentation about genomics. The field of genomics is very wide. Okay, it includes many topics. It includes many issues. Okay, and many perspectives sometimes also. Okay, we will see that there are many disciplines that take advantage of genomics. Okay, not only starting from the fundamental sciences, but also in applied sciences, to forensic, for example, sciences. Okay, and therefore there are many of these aspects that I will not include in my presentation. Okay, we will see a brief review of which are the sequencing technologies, and it is, I think, very nice to see also how sequencing technologies changed during the last 30 years, 40, 50 actually. Okay. And we will see that most of these technologies are not simply just outdated, but are still used, okay, with many limitations, but are still very useful in this field. Of course, I will mention sequencing approach and strategies, because these are important for knowing how to use genomics, maybe in your own field or application. Okay. We will talk, of course, about the issues, because as any field in science, there are many advantages. Okay? There are many interesting points. There are many interesting applications. Okay? As well as many backdrop, many pitfalls sometimes. Okay? Many difficulties also. Okay? And then we will talk about pipeline for analyzing the data. Well, I would say that this is not a very important part of this presentation, just because there are so many ways to analyze genomic data. Okay, it is impossible to be exhaustive in a single presentation, and it probably doesn't make particular sense to talk about everything about that, because there is a pipeline for analyzing whatever you can see within a genome. Okay, whatever variation, whatever divergence, whatever change in the genome, especially in the context of comparative genomics, that can be analyzed. That you may have tons of different scripts, software, pipelines, approaches, okay, to reach different results, different conclusions, okay? And often in this field, people working in genomics write their own scripts for analyzing the data, okay? So we will not mention any specific software or any specific pipeline, but we will see there are many different ways to view and see data points. Of course, we can't ignore the genome anatomy. What it is a genome, how is made a genome, what we can find within a genome, and which part of the genome can variable between different species, different population, even individuals. Okay, and then I will show you a few examples of possible genome comparison. A few case studies to show some of the most common application in comparative genomics. We'll say evolutionary biology. Why I stress particularly this point? First, because I'm an evolutionary biologist, therefore it's supposed to be this way. Okay, but really, if there is a discipline in biological science that is particularly grounded in evolutionary biology, is in fact genomics. The very same tools that you can use for analyzing genomes, even biomedical sciences, even in pathological sciences, okay, are all heavily grounded on the principle of evolutionary biology. Okay, for this particular lesson today, since we talk about comparative genomics, okay, well, we cannot avoid mentioning evolutionary biology because every comparison in biology, in science in general, but in biology is particularly important, okay? Every comparison is the foundation of every experiment we can do, of every observation. If we want to draw some conclusion from our observations, okay? Observing a simple phenomenon and not comparing that phenomenon in a different context or in a different biological systems, okay? It will not give enough information for understanding how it works in nature. Okay, so we have to compare. Okay, everything is comparison in biology. Okay, and in the very same moment you compare, you are doing evolutionary biology. Because evolutionary biology is, in fact, comparing things and understanding why things are different or similar. It is finding the driver, the principle, the reason why something, some structure, some specific tissue or some specific cell or cellular process changed, and why in this way and not in another way. Every time we talk about adaptation, every time we talk about speciation, every time we talk about divergence, migration, a population level or specific level or whatever it is, we are talking about evolution after all. And you will see, okay, there are many methods that are based on this principle. One of the most important principles in genomics and in evolutionary biology in general is the orthology conjecture. We will review the orthology conjecture later, okay, which is important to find genes across different genomes in order to compare the gene across multiple populations, multiple species, multiple lineages. Okay, well, the orthology conjecture came directly from the principle of homology, which is the base of the evolutionary biology. Okay, so that is for we will talk about biochemistry, medicine, ecology, physics, statistics, even informatics and mathematics, because as you may imagine, okay, when we talk about genomics, but probably with the presentation of today of Jeff, you already appreciate these points. We are talking about a huge quantity of data. Okay, and this huge quantity of data cannot be analyzed by hand. It needs to be analyzed with software, okay? And it is not possible to use a simple personal computer. We usually have huge, high-performing computer, server, okay, multiprocessor platform, whatever, because it may take time. Okay, therefore, informatics and mathematics are fundamental for doing genomics. Not that I am a mathematician, I'm not an informatician, of course, but we collaborate with them. Most of the model we use, most of the software we use have been designed by mathematician or statistician, or of course, informatician. Okay, and then the remaining of natural science realm of disciplines are all linked together in the evolutionary biology context. Well, genomics. We will see what is a genome. But I would like to start the story a little bit before to reach the genome. I would like to start from the technical point of view, because that should give you the idea, okay, of the development of the last 50 years in this field. Okay. Genomics, well, you probably know it's a matter of comparing DNA sequences. The difference between a simple DNA sequence comparison with respect to a genomic comparison, okay, is a matter of scale. A single DNA sequence could be 100, 1,000, few thousand base pair long, okay? A genome is a DNA sequence, which is millions or billions of nucleotides long, okay? How we can analyze all these data? Well, the principle of sequencing started exactly 50 years ago, okay, with so-called Sanger sequencing. Sanger was a guy, okay, which knowing how the DNA molecule was built and knowing how it replicates in vivo, okay, started to think about the methods for emulating this replication in vitro. Okay, you know that during the cell division, DNA replicates. Okay, this replication is made by using one strain of the double helix as a primer. Okay, and then it is copied. Okay, and we have a new copy of the chromosome or the DNA within a cell. Okay. Well, Sanger invented a way to replicate DNA exactly in the same way. And well, you can see that after 20 years, okay, we got the first genome sequencing using the Sanger method. And it was a virus sequence. It's a very small genome. But that was the first complete genome built. Okay, to give you an idea of the work that is behind, this should be a few thousands of base pair. It is not very large. It's smaller than a single gene of the human genome. Okay, to give an idea, okay, I started to work in this field in 2000, more or less, okay, using Sanger method, okay, in order to get a sequence 300 base pair long, okay, that is a very short, believe me, it's a very short sequence, okay, you need a couple of hours and a half using an automatic sequencer using Sanger method. Okay, so you can imagine how much time was needed to get a few thousands of base pair. Okay, so it was very important advances. Okay, and in the days, in the years later, okay, we got a lot of small genome sequences until more or less the 2000s with the first draft of the human genome. Okay. This first draft of the human genome took 10 years of work. Okay. And it was a very incomplete genome. And we will see what does it mean having a complete genome or an incomplete genome. Okay. And then things rapidly escalate. In the last few years, okay, we got so many genomes, okay, we don't even have the resources for analyzing all the genomes together. At this point of the sequencing story, the bottleneck was a technological bottleneck. It was 10 years for getting the genome sequence of the human that is 3 billion base pair. It's a 10 year. Now you need a couple of hours to get the same genome, the same level of coverage. Okay, that is to give you an idea on how fast this field developed in the last 10 years. Okay, now we are 5 years later than this date. Okay, the IRGA consortium is a European consortium aiming to sequence every organism in Europe at chromosome level. Okay, which is at the moment the top level genome sequences we can get. Okay, and by the time we are talking, there are several thousand of mammalian genome sequenced. Okay, and we have several hundred of thousands of animal genomes sequenced. Okay, most of them, 90% of these in the last 10 years. Okay, these are all consortium that in the last 10 years started to promote genome sequencing. Okay, the IRGA is just the last one. Okay, but most started with the Earth Biogenome projects a few years ago and the Vertebrate Genome projects and then before. Okay, so this is a field which is rapidly developing. Why I'm talking to you this? Why do you want to care about this? Because these consortium, okay, are just interested in producing new genomes. Having genomes for all the species we know, okay, means to have some information about all the species we know. Much of the species we know presently, okay, we only know morphology, something about their distribution. For the majority of them, but not all of them, we know the phylogenetic position in the tree of life. Most of them, we don't even know nothing about every possible specific cellular process. That's the fun of biodiversity. We have so many different species. Okay? We don't even know how they work. We don't even know how they integrate in the ecological network. We know that they are there. Okay, so having a genome sequence for everything is a step forward in the understanding of their evolution and for their ecology and their relationship with us, with our activities. So what could be our interest in knowing that there are so many people that are eager to sequence everything is that they do, if it is an interesting story, okay, to sequence in your study. Do you need to sequence some strange fish which is important for something? You may ask, if it is a European species, to Andrea, if they produce in your stead a genome, chromosome level genome, which is like having a Ferrari currently, okay, then you can use in your research. Okay. The amount of genomics, okay, doesn't require to be expert cleaner. There are many people that can clean that for you and give you the name of the mount. Okay. Okay, so you can exploit all these opportunities to have something more, something that could be useful. Okay, maybe for many of us, it's not important to have a new genome because that genome is already sequenced, or we are not interested in genomics at all. But if you think about what you can do with a new tool, knowing genome sequence, okay, is just a new tool for us. Is some more information about the biology of that given lineage, that given species, that given population. Okay, therefore, why not ask for that? Okay, and see what we can get from that genome. So, there are so many consortia that works in this field. It is very likely that every organism is covered by one of them. Okay, still a little bit of story. This is the timeline on sequencing developing, and here is the Sanger sequencing. We are basically here, is a little bit outdated here. And we are still using technologies that are being developed in the last 10 or 15 years, okay, say the upgrades, okay. What they would like you to appreciate, okay, is not only the fact that it is very easy currently to get a new genome, okay. I say that getting a new genome sequence for Homo sapiens, for example, it will take a couple of hours. That is true, in fact, okay. But this also because we know perfectly how is the genome of Homo sapiens, because we are now waiting for the 40 version of the human genome. Okay, starting from 2000, okay, when the first draft, that is version number 1 of the human genome has been issued, okay, we are at the 39th version and waiting for the issue of the 40th version, okay. There are some regions of the genome that are very hard to sequence, okay. Some say that it's impossible to sequence everything, but we are approaching the perfection from this point of view. In many apes, okay, we already have all chromosomes from one hand to the other end fully sequenced. One thing that it is still lacking in the human genome. Okay, therefore we know a lot about assembling a human genome. Maybe we don't know exactly how to assemble the genome of fish species or a mollusk species or whatever organism species that have not been sequenced before. Okay. Therefore, it is not matter of a couple of hours. Okay, it could be a matter of a couple of months. But if you work focused on that genome, okay, in a couple of months, it is possible to have a chromosome level genome for every organism. To give you a couple of time in this field, okay, running the real sequencing, that is getting the raw data, okay, could means waiting probably a couple of days. Okay, the automatic sequencer work for a few hours, okay, but data need to be prepared, library need to be built, okay, everything need to be checked and set at the state of the art, okay, and then, okay, sequencing, okay. It's very easy getting the sequencing, the real sequencing, okay. When I was a student, 300 base pair, 2 hours and a half. 2 hours and a half, probably now you get billions, okay, of base pair. The bottleneck is no more in getting sequences, the bottleneck is in analyzing sequences, okay. We're going to see how to assemble a genome, okay, and it is a fucking mess, okay. It could be very, very hard. If you don't know nothing, if you don't have already a draft that can drive the analysis, okay, it will be a jump in the void, okay. You know when you start, you may know not when it is finished. Typically, a couple of months are necessary to have a first assembly. Often, people working on assembling genome doesn't work only on a single genome. Okay, so things there's a little stratification of work that can take the 4 months. Okay, we are in my research group. We are sequencing a couple of genomes chromosome level within the IRGA consortium. Okay, they are still working on that since say May, April, May. Everything started that before. We are still waiting for getting the genome set. Okay, so this is one point that have to be taken into account, especially when you want to lay down a new project, okay, and you foresee to sequence some new genome that could make a reference genome. Could be the base of your real analysis. Okay, the tricky point is that getting simply the genome is just the starting point of everything. Okay, you work a lot for having just the starting point of the whole story. Okay, then there is all the remaining of the study. The point is that how far we can go with sequencing. Okay, you may have a little perspective on these. Okay, looking at this table here, where are listed, okay, some of the sequencing method. That is the first generation method, is the Sanger sequencing. Okay, and we will going to spend a little few moments making the difference between Sanger and the remaining of the other methods. Okay, then there is the second generation methods here. Okay, and then we are now exploiting the third generation methods for sequencing. Okay, and you can see. Okay, look at this column here, read length. It is how long could be the sequencing using that technology. Okay, how long is the human genome? Human genome is 3 billion base pair long. Okay, it is a single sequence. No, yes, 23 different single sequences. It is the 23 chromosomes. Okay, every chromosomes could be several millions of base pair. Okay, so it is not possible to have a single sequencing T to T, let's say the telomere to telomere, is here from one end to the other end of the chromosome. Okay, there is a limitation. That limitation, better known as the second principle of thermodynamics, means that at a certain point, okay, the energy is sufficient for getting the end. Okay, so all these technologies are characterized by different lengths in sequencing. So the Sanger method has its own limit to 1,200 base pair. It's a very small DNA sequence. In certain cases, is like 1/10 of a single gene. Okay, this is very small. Just me. Why 10 years for getting the first draft of the human genome? Because it has been used the Sanger method. 3 billion base pair covered by small fragments. Okay, 1,200 base pair long. Okay, it is the maximum efficiency. Okay, the average is to 900 base pair. Okay, and for every read, okay, you need like a few hours. Okay. When moving to the new generation sequencer, okay, you can see that the length of the read here decrease, very substantial extent. Okay, it is not listed here. The most, it is here. It's HiSeq and MiSeq. These are the so-called Illumina sequencing. Okay, and which I'm sure you already know. Okay, Illumina series is the most widely used method for sequencing. It's very cheap. Okay, it produces rates that are on average, usually are 150 base pair. But within a single hour running, okay, of these 150 base pair, you have billions of sequences. Okay, we will see how it is possible to cover the whole genome like this way, but the point is that reducing the length of the reads, okay, but increasing the quantity of the data, okay, you can still get more easily, probably, okay, a single genome, okay. The point is that it's very easy to get billions of data in this way, okay. It's not easy to assemble everything together. That's why we use months for getting the assembly of the genome. Last methods that are going to be the new standard for having the genome sequences, okay, are the PacBio and the Oxford Nanopore, okay, which produce very large rates. When large means 20,000 or a few 100,000 base pair long. Okay, still you need a lot of them. Okay, so that is for giving an idea, okay, on how things work in this field. From a technological point of view, we are not really interested in how many time we need to get those data or how long reads are. Important for us is to getting the genome, okay? It will be probably an issue that some technicians somewhere need to deal with, okay? What we get at the end of the story, okay, is just the genome sequencing, okay? But of course, these could be important, okay, when analyzing that genome, knowing how that genome has been assembled. Assembling genome with only short reads could mean that you lose something because the way short reads are assembled, we're going to see that. Okay. It is better, that's why it's the new standard, it's going to be the new standard. It is better to have thousands of reads, but very long. Okay, because let's say the structure, the architecture of the genome, okay, is rebuilt during the assembly more easily. But it is unfortunately much more expensive. Okay, so sometimes we have to find the sweet spot in between. These are the guys that work for us. These are the sequencer. Okay, that was the old science sequencing, and now this is the Oxford Nanopore, the mini ion. It's very interesting because it's large, like a smartphone. Okay, and you can bring with you everywhere. Okay, it's very easy to use. It is also not particularly expensive. The machine, it is around €1,000. Okay, but it is very expensive making it work because the chemistry is very expensive. This is another view to see where, with respect to the read length and the throughput, it is how a single run output in terms of base pair, okay, is working. You can see that PacBio, which is the new standard, the one using very large reads, okay, it is a very large output for a single reads, but you can produce in a single run just 1 gigabase. It is 1 billion of base pair. Okay. The Illumina family, which is here, these are all variants of the Illumina reads. Okay, very short read length, but a very large output. Okay, and you may say that what's the point of maintaining a very large output but very short reads that are also tricky to assemble? Okay. Well, the fact is that first, you have so many data, okay, that even if the reads are so short, when assembling, okay, you have so much data that you can assemble confidently all the genome without error. Hmm? The truth is that we are still working with these because it may be used for re-sequencing. If you want to analyze several individual, several population, okay, in population genomics, forensic studies, okay, you have a one reference genome, maybe built with the PacBio reads, okay, very strong structure, very large chromosome level, maybe, okay, and then resequencing with this very cheap lot of data, okay, you can have a very deep sequencing and very deep knowledge about alleles or genetic variants that you may be interested in. Okay, just to give you a comparison, the Sanger sequencing is here. So you can see the output. It's very small compared with that. Okay, now how things works. Okay, well, that is the moment to talk a little bit about Sanger sequencing on one side, okay, and the next generation sequencing on the other side. There are advantages in working with Sanger sequencing. Sanger sequencing, just to say, it's a reasonable length of the read, okay, and the throughput very small. That means that you have to wait lots of time, a lot of work, okay, a lot of wall time. It is a lot of machine working actively for getting you the same quantity of data as Illumina sequencing or even a PacBio sequencing, okay, but the Sanger sequences is a direct sequencing of the molecule. It's a real chemical reaction that give you exactly the sequence. The Sanger sequencing is still used for validating some chromosomal regions that are difficult to assemble or maybe artifactual, okay, by using next generation sequencing. Okay. It's like that when you have Sanger sequences, you can be 100% sure that is the real sequence you get from the genome. And it could be 1,000 base pair long. If you take 10 reads, okay, from Illumina sequences, because they are 100 or 150 base pair long, okay, in order to get the same length, you have to assemble in the largest DNA sequence. Okay, and that's the moment when you realize what you're doing, okay, is based on statistics. Who say that those 4, 5, 6 short reads should be assembled that way? A mathematical model which take all your reads, assemble them together, and say that that is the best way to assemble that sequence, giving you a score. Okay, I can say it is wrong. 99% of instances, okay, it works perfectly, finally. Okay, still, if you find some which doesn't look sound, or doesn't work, or it is waived this way, okay, you can be only sure about what you are looking, okay, by Sanger sequencing the same readings. But I said, assembling even a single chromosome by Sanger sequence is very hard. Mm? Really expensive, not really efficient, let's say. Short reads. Start talking about short reads, because short reads was the standard in the genome sequencing until the last 5 years. 5 years, 6 years ago, more or less, okay, PacBio and Oxford Nanopore started to become a popular way, okay, to sequence genomes. Okay, before that, okay, most of the genomes, which are in fact currently sequenced using long reads, okay, were only assembled using short reads. Now imagine to have a story. No, well, we'll see later. Imagine to have a puzzle, a jigsaw, you know, only with 1 million of pieces. Okay. You have to start assembling by hands, okay, looking if they match, they do not match, the color, the figure, okay, shape, whatever, okay. It takes time, okay. If you are a supercomputer, okay, well, it will take less time, but nonetheless, okay, it will take time, okay. Why? Because you have a huge population of short reads, okay, and you have to put them in the correct order, so that when you have all of them collapsed, every of them on each other, you get finally a largest sequence, which is your chromosome. Okay, when using short reads, at the beginning we got only single-end sequencing. It means that because the method, I will not go too much in detail, but we'll say the method is very simple. You get a genome from the cell, from tissue, okay? You put them in a solution, okay? And then you trim the genome in small fragments, okay? Very small fragments, okay? And then you sequence just one end of the fragment, which could be 100, 150, 200 base per long, okay? Another way is to get all those fragments, maybe not so small, or better, some very small, some very large, okay, and sequencing both ends of each fragment. So you have the sequence of the first 100 base pair of one fragment, the sequence of the last 100 base pair of the same sequence, okay. You still have two short reads, okay, but you know that they are linked to each other by an unknown sequence of some length based on how you share the genome. Because you can select how to share. It is possible to fragment a genome with fragments all 350 base per long, all 550 base per long, 800 base per long. So you know how each of these fragments are long. Okay. But still, the method is to ascend all of them in order to get a larger sequence, which is named content. Well, that's exactly what I'm saying. That is the fragment. You have the read one, you have the read two, okay, and you know which is the relative position to each other, okay. So you know that those fragments you know only the beginning and the end, okay, could be some way ordered across, for example, a reference genome, if you are comparing different genomes, for example. Long reads are sequenced in a slightly different ways, okay. Here is just to show you the way the molecule is treated during the sequencing, okay. And well, it is very interesting because while the scientific sequence is the chemical reaction, okay, a real chemical reaction, okay, and you reconstructed the sequence by observing the product of this chemical reaction, okay, all those sequences are indirect knowledge of the sequence because they use the fluorescence, which is emitted by specific nucleotides, okay, during the read of the sequencer, or even the change in the current, okay, within the solution, okay, because every nucleotide has its own charge, electrical charge. Okay, so by the way, and this, of course, leads to have a more efficient sequencing. Therefore, you can get larger rates. What to do when you have your sequencing? Okay, this is the most delicate part of the story because assembling the genome, let's say, is currently the real bottleneck of the story. It is the moment you spend much more time, okay, in order to have proper assemblage. Okay, there are several different methods, several different streets, software, pipelines, okay, which allow us for assembling based on kind of sequencing. It's short reads or longer reads, okay, or the use of a little more refined methods for getting data. The raw reads are the product of the sequencing, okay, which is usually made by a company. Running the next generation sequencer is very expensive, okay. It is very difficult that a single laboratory can have its own next generation sequencer. Because it is matter, it's a financial point of view here. Okay, if you want to have a decent efficiency of your sequencer, that sequencer have to work 24 hours per day, 365 days per year. Okay, otherwise you are lose money, time, work. Okay, so that's why 90% of instances people prefer to pay companies which have these facilities and work pretty well. Okay, and only few universities have their own sequencing center. Okay. The nearest to the University of Bologna, for example, is in Florence. The University of Florence is a mega hotspot, okay, and there's its own sequencing facility. In Italy, you have the Human Technopole, you have the University of Florence, the University of Bari, and then private companies, don't say. Therefore, it's very, because it's very expensive to maintain all the machine running. Okay, so the raw reads is your basic data. Okay, and then you have to know what to do with them. Okay, and this is the first, dichotomy in the workflow. Okay, why are you sequencing the genome? Okay, are you doing a new genome? Are you sequencing a new genome? Do you need to assemble a genome that has been never assembled before? Okay, well, you are doing de novo sequencing. Imagine species of an organism. Okay, at this very moment, it is very likely that, for example, most of endemic species everywhere in the world, okay, still waiting for having their genome sequenced. Okay. I'll say the novel sequencing, okay, is sequencing something new, something that has not been already sequenced, maybe even sequenced in a very loose way, in a very having a draft which is rubbish, okay, but still you don't have any idea of what to find in the genome, okay. De novo sequencing, okay, currently means in the last 5 years and likely in the next few years, okay, is made by long reads that is producing a lot of very large reads like PacBio or Oxford Nanopore, okay, to have the basic assembly, and then the IC mapping, we will talk about that later, okay, which is an additional technique, okay, which allow to get the chromosome sequence. That's the usual, the well-known chromosome level assemblies are made only after the IC technique, okay? And then you have your de novo assembly, okay, which, as said, is just the starting point of your story. The other side, you are sequencing something that is well known. You are an anthropologist, you are a forensic scientist, for example, and you want to know a few guys, maybe criminals, or maybe a new population of indigenous people lost in the middle of the Amazonas, and you want to know, okay, which is their ancestry, which is the origin, the ancestral origin of that population. It is what most of the evolutionary anthropologists do right now. Okay, we know already the sequence of the human genome. Okay, it is one of the probably not the most detailed or the most complete genome we know currently, but we know a lot about the human genome. This makes no sense to make a new de novo sequencing of the human genome. So we can use the short reads in order to resequence. Resequencing, okay, is doing sequencing experiment typically with short reads. Why? Because it's cheaper. You can do resequencing also with PacBio reads, of course. Not cheap so much, okay? Especially if you think to perform this experiment on multiple entries, like several populations, several guys, several conditions, like in medical science, for example. In cancer science, where sequencing is very common, okay, in order to get the genetic variants that could be linked to the pathologies, okay? And therefore, the use of short reads allow you to resequence, okay? And then all those short reads are mapped on the reference genome, which is the 39th version currently of the Homo sapiens genome, okay? And at the end of the story, you get something that, like in de novo assembly, allow you to identify genetic variants, identify genes, which may be surprising, but every time we sequence new species, we found new genes that are only present in those species, because species differ also by the number of genes. Very interesting part, we'll talk about that, functional annotation. Finding genes, well, fine, you find a gene. What's the task of that gene itself? Well, you know, functional annotation is an entirely statistical pipeline based on what we know about one of the most, about one lot of the most known genome, like the human, like the Drosophila, like the C. elegans genome, okay? And those functions are linked to that gene, okay? And can help us, okay, in understanding what could be potentially, okay, the function of those particular genes. We'll see later why it is interesting in that. Okay, and then you have your results. Okay, that's a typical pipeline. The point, only relevant point, of course, is where you go from this point, de novo sequencing or resequencing and then mapping. Well, I don't know why I put this slide here, but it's very nice. Okay, that's a typical shape of the output of the sequencer. Okay, in all these mess of characters, okay, these are the DNA sequences. Okay, you see, and for every DNA sequence, we have additional lines here, okay, which include different information which are not useful for humans. We can read here and we can see a lot of character, okay, without any real meaning for us. It's not, for example, this, which is the name of the line. But these are important to univocally identify the reads, okay, but more importantly, okay, it is important this line here, okay, because tell you something about the quality of the sequence. This is what the sequencer reads on the DNA molecule you put within the input sequencing, okay? And all these letters here tell you how confident we can be about the presence of a T in this position, the presence of a G in this position. That is important. That is important because the efficiency, okay, is never the 100%. It is important because when you sequence, okay, every change, maybe in the temperature of the lab, maybe jump in the grid, and therefore you have a glitch in the current within the machine or something, can produce an error. And if we have an error, okay, we cannot be confident about the results of the run, okay, in which the error occurred. So if in this point something occurred, okay, that produced an error, the corresponding A here, okay, could not be sure 100% an A, okay. You put an A because something telling me that there is an A here, okay. But the quality is telling us that the quality is not very high. Okay, so it is possible that that A actually is not an A, okay? And therefore, first things to do when getting the raw data is clean the raw data. Raw data have to be cleaned by a lot of stuff, including byproducts of the reaction, of the sequencing reaction, okay? And that is a classical view of the distribution of the quality. Okay, this is a 100 base pair long reads ISAC 2000 Illumina. Okay, single end. Okay, and you can see that the first few position, okay, is a very high quality. These are pretty sure. For some reason, going to the last part of the sequence, okay, you lose confidence. Okay, that is actually a typical pattern. Okay, the last are always less confident than the first position rates. Okay, the difference could be where these medium lay. Okay, often when you have a high quality read, okay, even the last position, okay, it is still worse than the first position, but you have this signal on this position here more probably. Okay, this is particularly bad sequencing. Okay, that is very important. Many scripts or softwares have been produced, okay, developed specifically for cleaning the reads from errors. Okay, and what's the point of getting short reads but getting billions of these short reads? Is that when you assemble like these, if you have an error on these reads, okay, it is unlikely that this position is wrong also on these and on these reads. Okay, therefore, if you have many reads covering exactly the same position of the chromosome, okay, you can statistically reconstruct which is the most likely position. So you have two ways to do that. Okay, you have one or few reads having errors, okay, and the majority of the reads having the correct nucleotide, okay. When piling up during the assembly, okay, in the resulting sequence, you have the correct position. Either way, there are no errors at all. Therefore, you have your position correct. Okay. What we can do to facilitate this process, okay, is to run some read cleaning scripts, okay, which eliminate those positions. But for example, which can be useful for eliminating all those positions was quality score goes below 30. Because we say arbitrarily set our quality threshold to 30, okay? And therefore, below that threshold, we say that we are not confident anymore that that position is correctly read, okay? Therefore, it is cut out. Getting rid of that, okay? What it remains, okay, are all reads that in those positions have the correct reading. Okay. One of the most common methods that you may read about is, for example, Trimomatic. Trimomatic is a pipeline, okay, which can be installed on a computer, needs a little bit for working, okay, a huge of RAM, memory of the computer, okay. Does that will result, okay, in a new set of reads, okay, that if you compare with the raw reads, that is the output of the sequencer, you can see that the Trimomatic produced a smaller set of reads. Because getting the threshold as a new position to work, okay, most of the reads, many reads can be directly eliminated. How to assess them? Well, that is a very important point that can be summarized with this sketch here. So imagine to have, okay, a few newspaper, okay, all identical. This is the same copy of the same newspaper, everything piled up, okay. Then you make them exploding in very small fragments, okay. And then take all those fragments and reconstruct a single copy of the newspaper, okay. That is the de novo assembly, okay. This is your genome, okay. You shear your genome into small fragments, okay. You sequence them and you get a lot of very small pieces, very short reads. But even long reads is the same story. You have different reads. Most of them are redundant because you put in your basket several copies of the same newspaper. In the same way, when you sequence your genome, in the solution, the DNA solution you put into the sequencer, there are several copies of the same genome. The DNA is isolated by tissues. Tissues is made by multiple cells, okay? And in these multiple cells, therefore, you have multiple copies of your genome, okay? That is the shearing, okay? And then the reconstruction, that is the de novo assembled genome. So you see your more serious slides, okay? You see multiple genome copies. That you can imagine that every of this line is a genome, okay? These are isolated from the nucleus of different cells of the same tissue, of the same individual, hopefully, okay? Then you produce your reads, and your reads are very small fragments of all these, okay? These are all identical to each other, being copies of the same genome, of course, or in principle should be identical. But let's not complicate too much this thing, okay? Therefore, you get your reads here, okay? And fragment assembly is to make order, to put all these reads exactly in the correct order in order to reconstruct a single line here, which is hopefully identical to each of your original sample. And, well, you may imagine that there are a huge mass of mathematical models, okay, that help to put the reads all together, okay, to get that sequence. Okay, these open a course of several days talking about of mathematics, which I'm not able to do, by the way, therefore, trust me. Okay. It is not important the way you get the model you use for assembling the genome. The only important point, okay, the only tip you have to care about, okay, is that the method you are using is suitable for the kind of sequences, short reads, long reads, okay, and it is suitable for the kind of genome, which is a little bit more complicated. Not all genomes are identical, of course. Genomes are all different. Even when comparing different individuals of the same species, but we can extend this, saying that in our very body, every cell doesn't have exactly the same genome of the other cells. Okay, because there are variants everywhere. Okay, therefore there are not such a thing like identical copies of the genome. Okay, and that's not the point. Okay, the point is that some genomes, okay, are more difficult to be assembled, okay, because they are composed differently with respect to other genomes. There are genomes that are so huge, okay, it is currently technically impossible to be assembled properly. The human genome is 3 billion base pair long. There are some amphibians, okay, that have genomes that are tens of billions of base pair long, or hundreds of billions of base pair long. There are some organisms that have more than 100 chromosomes. Okay, there is so much data that even a supercomputer can calculate for years, probably, before to reach the saturation of the memory. Okay, so there are many organisms that have been sequenced, yes, but their genome is not assembled yet, because it is still lacking the computational power for doing that. Okay, there are genomes that are full of repeats, that are repetitive DNA sequences, like transposable element or other kind of repeats, which are some kind of DNA very small or relatively small DNA sequences that are repeated even thousands or millions of times within the genome. Why we are not able to sequence, for example, human centromeres? You know what is a centromere? The centromere is where every chromosome is attached to the mitotic spindle when they are take apart during the cell division. Okay. Well, in the human genome, every centromere is made by so-called satellite DNA. Okay, satellite DNA is a cluster of 170 base pair long sequence, which is repeated ahead of time several thousands or several millions of times. Okay, it is impossible to pass over a single centromere even with long reads. Okay, the problem is that when you sequence a small part of centromere, you don't know how to assemble it. Okay, because it's made of repeats, so it can be positioned in several ways. Okay, and you don't know which is the correct way. There are some genomes that this problem, okay, is amplified because the repeats are distributed across the whole length of the chromosome. Okay, therefore are very difficult to be assembled. Okay, therefore in these cases, okay, it is not irrelevant which model, which algorithm you use for assembling, because some algorithms are actually designed specifically for dealing with repetitive DNA. Okay, therefore, a well-known, full of repeat genome assembled with an assembler using an algorithm which is designed for assembling the human genome, okay, likely it is not properly assembled. Okay, but that's the only point you have to take into account. This is, as usual, to use the correct tool, okay, for the task you are doing. Otherwise, every tool is okay. Difference between short reads and long reads when assembling. Okay. Okay. Assembling a genome doesn't always lead to get the 100% of the genome reconstructed. We mentioned right now the problem of the centromere, which is not a problem of the human genome, but is a problem which is common to many different genomes. Okay, we are just in the last years, thanks to some long reads and some specific, particular, still experimental model, okay, assembling telomere, centromeres, for example, in the rice, okay, or in the gorilla or the orangutan, which have very small centromeres. Okay. But generally speaking, often when you get the 85% of the genome sequenced, okay, that is a very nice sequencing. Okay. But in that 15% of the genome that has not been sequenced, that is assembled, okay, there are not only centromeres, there are not only telomeres, which are also full of repeats. There could be also parts, functional parts of the genome. Okay, so you can miss some gene, for example. Well, this is frequent, okay, when assembling genome with short reads. With short reads, okay, often, we know now, okay, often lead to incomplete genomes, okay, because of the nature of the assembly with short reads. Long reads, actually, okay, are so long that if you produce enough long reads, you are pretty confident that the whole length of the chromosomes are covered. Okay. Reconstructing is a different thing. Okay. Recovering the whole length of the chromosomes, okay, it would be easier with long reads. That's why currently Producing de novo assembly of genomes without long reads, okay, is not more acceptable. Okay? If you now try to publish a new genome, a de novo assembly genome, only using short reads, okay, well, people say, reasonably and correctly, okay, that is not a genome sequencing. That is at the best, it's a genome survey, where you can be confident that more than half of the genome is covered. Okay, so that's why it is important to use long reads. That is a assembly in brief. Okay, let's imagine to have short reads, for example. Okay, all those short reads are ordered based on their overlapping regions. Okay, question could be, how can we be sure having very small short reads, okay, that every read have an overlapping with another one? Okay, well, the answer is that if you produce 10 short reads, it's very likely they do not overlap. If you produce 10 billions of short reads, an overlapping somewhere will occur. Okay, the overlapping is when two ends match with each other. Okay, probably with some difference. Okay, which can be, of course, produced by error. Okay, or could be a genuine polymorphism. Okay, maybe there are different alleles you are combining together. Okay, but it is acceptable. Okay, and this overlap of two reads produce a larger one, which name contig. Okay, these contigs are then put together using mate pairs, that is, using short reads that are on one end of a fragment and another end of the fragment, which link each contig together. It's like a second phase assembly. First phase, assembling the reads into contig. Second phase, assembling contig into scaffold. Okay. Scaffold are those that are sequence that we get at the end of the whole process of de novo assembly. Okay, and these scaffolds could be chromosomes if we use also the IC method. Okay, the ultimate scaffold is the chromosome. The aim of every chromosome scale sequencing project, okay, is to get as many DNA sequences as the number of chromosomes. In the best case, okay, in the expectation we all want to have at the end of the process, okay, imagine to sequence the human chromosomes, which are 23, but in every cell you have 46 chromosomes because we are diploid, okay, is to have 46 scaffolds. One chromosome and its sister chromosome. Okay, that's the ultimate sequencing. Okay, success rate 90%. It's very difficult to get only the sequence of the chromosomes, but that's the aim of the chromosome scale sequencing. Okay, before the end of the story is that from longer short reads, sometimes also mixing up both long and short reads together, okay, you can get scaffolds. In the best case, if you use the IC methods, which is depicted in this figure, which is a way to capture, okay, the positioning of the DNA sequencing within the nucleus, okay, using some enzymatic activities here, okay, and then the resequencing based on their positioning in the nucleus, okay, we can be able to get this scheme here, where each square represents a chromosome. Each square along the diagonal. Okay, this is one chromosome, this is another chromosome, this is another chromosome, and so on and so on. It's not important to go into detail here in this case, okay? But let's imagine that the AC is currently the only method that allows us, okay, to build a chromosome level genome. Okay, if you read a paper, if you are allowed to use a genome, okay, which is indicated as a chromosome scale, okay, but you know people don't use the IC method, okay, well, that's not a chromosome scale. That's a highly contiguous, highly accurate genome sequencing, okay, which could be fair enough, could be enough, okay, for getting the information you need in your project, okay, but not a chromosome scale. The chromosome scale is a different thing. The chromosome scale could be obtained only by the IC methods and similar methods that are still in use in different conditions. How to judge whether a genome is assembled properly? We say that assembling a genome is a matter of getting a billion of pieces of a jigsaw, okay, and try to match them together, okay, based on scores, basically. Okay, every algorithm take the reads, short and long reads, whatever it is, okay, and assemble together, looking for matches and mismatches, okay, and then outputting, okay, scaffolds that are characterized by a number of statistics, okay. Based on these statistics, we should be able, okay, to evaluate whether our final output of the analysis, okay, is a suitable result. That is, we can use it for doing all the remaining of the analysis, okay, or we have to make some more analysis. We have to, sometimes we can realize that we need to sequence more to get better results. Or we have completely to change, okay, the method. Okay. We can easily realize, okay, that if we use only short reads, we will have a genome, okay, which could be suitable for something, okay, but whose statistics is for sure worse than same genome sequenced with long reads. Okay. Typically, the basic information of we look for when evaluating a genome, okay, is a single statistics which is named N50. Okay, the N50 is basically the number of scaffolds, okay, that are larger than the median length of the all scaffolds within the genome. Okay, the larger the N50 score, the better our genome. Okay, that is, for example, the statistics of I don't know which genome it is, but it's a genome. Okay, after the sequencing, we got 300,000 scaffolds. It's a very fragmented genome. Okay, imagine that could be, that is, it's a 2 gigabases, it's a 2 billion genome sites. Okay, 2 billions, what could be? 2 billions could be probably, I think the gorilla or the orangutan is about 2 billion base pair. Okay, imagine to have that genome, okay, fragmented in 300,000 scaffolds. The number of chromosomes of the gorilla is around 20. Okay, very fair enough. If we got a chromosome level, we should have, if not 20 scaffolds, 30, 40 scaffolds could be nice to get it. Okay, the M50 tell us that the number of scaffolds of these 300, okay, the median above which alpha of the scaffolds, okay, is larger, is 12,000. 12,000 is the median length of a single gene in a mammalian genome. It's very small. Okay, so it is likely, okay, that in this number of scaffolds, okay, we have many fragmented genes. Okay, that is, we have only a partial information of most of the genes that are within this genome. Okay, that's the picture, okay, of a sequencing experiment that needed to be redone better, or an assembly which is better to rerun again, okay, in order to get better results. Now, to give you a practical example of what it is a difference of sequencing short reads and long reads of the very same genome, okay, that is an insect, so it's a Japanese termite, okay, and the genome of Japanese termites have been sequenced twice. One with short reads and one with long reads. The resulting genome size, the length in terms of base pair of the assembly is basically the same. Here is an approximation to the round up, but the number is the same. Okay, so we got two genomes of exactly the same length. We have one order of magnitude more scaffolds using short reads. Okay, the M50 is lower when using short reads of one order of magnitude, because these are 37 kilobases. These are 37,000 base pair. Okay, and here the M50 is an order of millions of base pair. For the median length of 1/2 of the 400 scaffolds is larger than 44 million base pair, which is much more than the other. And then we have the genome coverage here, which is completely another story, but the genome coverage is how much every single position, nucleotidic position in the DNA sequence of the assembly is covered by a number of reads. In this case, 59 reads cover on average every position. Okay. In these experiments, not only we have larger reads, long reads, therefore, leading to less scaffold and then a better M50, okay, but we also have the double of the genome coverage. And it is every single position is covered by, on average, by 114 reads. Okay. Therefore, it is more contiguous, each scaffold is longer, okay, and it is also more accurate. The higher the coverage, more accurate, okay, is the DNA sequence resulting from the experiment. Okay. Often, when we sequence de novo, okay, a genome, okay, it means that we know nothing about the genome of that species. Here it is easy to compare and say which is the best assembly here. Okay, why? Because these two experiments sequenced exactly the same genome, okay, resulting in exactly the same genome length, in fact, okay, but we can say that, well, for sure, the PacBio sequencing is better for assembling the genome. If I have to use that genome for a new experiment, I will select this one, not this one, of course. But sometimes you don't know nothing about that because it's the first time you analyze that genome. It's the first time that genome is sequenced. Okay. If I have an assembly, okay, which is 1 billion base pair long, okay, which part of the genome is sequenced? If the genome of the sample I'm going to analyze is, for example, a human sample, which is 3 billion base pair long. Okay. If my sequencing experiment results in 1 billion sequence lengths, okay, then in the best of the case, I realize the sequencing of only 1/3. 30% of the genome sequencing, right? Okay, but who say which is the length, the genome size of my species? Okay, that is a very tricky information to get often. There is an experimental way to establish the genome size of the organisms. Okay, it is by analyzing through the coloration of the cell nuclei, okay, with some color like a Ferg. This is a Fergen coloration, but there are also other methods, okay, that allows us to estimate the genome size of every cell. So that if you want to know how large is the genome of a given organism, you have to isolate a few cells, okay, used with the Fergen coloration or flow cytometry, whatever it is, because we know that the volume of cell is proportional to the gene size. It's also named nucleotypic effect. Okay? The larger the genome size, the more space the genome needs within the nucleus, okay, the larger is the cell. Comparing with a known genome size, the known genome volume, cell volume, like, for example, Drosophila, and comparing the volumes, okay, we can estimate the genome size, okay, independently from the assembly. Okay, these are a number of genome assemblies, okay, which are the results of very accurate sequencing experiment. Okay, and you can see, okay, that when things are properly done, okay, the assembly size, okay, is strictly proportional to the genome size estimated experimentally. Okay, and that could give you an idea, okay, on how good is your experiment. Okay, but not always things work like that. Look here. This is a solid violation of this correlation, okay, in which estimating genome size is larger than the assembly size. That could be the human genome, for example. But we still have large holes within the chromosomes. Okay, that means that some part of the genome has not been sequenced yet. We miss something. Okay. In this case, we have to establish if we can accept this gap in our assembly or not. Sometimes the reverse occurs. We have a larger assembly size than the estimated genome size. Okay. Apparently, this means that we sequenced more than expected. How is that possible? Well, it is possible in actually several instances, like having contaminant organisms. This actually may happen quite frequently, I would say, if you squash an organism completely, because maybe it's very small, so it is not possible, for example, to eliminate the gut. Well, when you sequence the DNA collected from that sample, okay, there is not only the genome of the organ, but there are also the genome of the microbiome, for example. So you assemble more than the genome of that organ, resulting in a larger genome size. Okay, that would be the case to investigate okay, the nature of different scaffolds in your assembly sites. You may realize that you assembled not a single genome, okay, but 4 or 5 different genomes. The organism you are going to analyze, okay, the one that is the reason of the experiment, okay, and for example, many symbionts, gut symbionts, which are very common. Okay. That is possible, okay, or a frequent issue when using very deep sequencing with longer reads, okay, is to assemble sister chromosomes. Okay? So you assemble more than expected because you assemble 2 Amy genes. So you have to say that you have 2 phases, 2 alleles. Okay, so you get the double of the length because many of these are exactly the same region, okay, but are not assembled together because there are many mutations that produce the gene variants, okay, so these are assembled separately, okay, resulting in 2 copies of the same genomic region. That's why you may have a larger assembly than expected. 2 minutes, then we have a break. Okay, just to show you the last before the coffee, okay. Assembly quality cannot be assessed only based on the comparison with an estimated genome, okay, for 2 reasons. First, often, unfortunately, you don't have the information of the estimated genome size because doing flow cytometry or doing foreign coloration with the single cells is not that easy. And often it's a matter of money and time, expertise. Okay, so some organisms are also difficult to track in these conditions. Therefore, not always we can get that information. And I would say that not always is important to get that information. Okay. The most important part of the genome, sorry, that's not true. It is not the most important part of the genome. It is the part of the genome we are interested in more. Are the genes. In the majority of experiments, in the majority of the projects, which include genome sequencing, okay, what we are interested in is to get the sequence of the genes of the genome, because that's a functional part of the genome. Analyzing those genes can get us better information about cellular process, tissue function, local adaptation, evolution, whatever. Often it is better, for example, to make a transcriptome approach rather than a genome approach, because with a transcriptome approach, you directly have the gene information and the effect on gene expression based on different treatment. Okay, you miss part of the story, but it is more focused on that topic. Therefore, it is not important to get the 100% of the genome assembled. The most important part for most of the experiments is to get all genes assembled properly. Because comparing and analyzing those genes, we can get more information about basic biology of the organism or the effect of the organism on the environment and vice versa. Okay? So we have to use a way to estimate whether in our sequencing we got an acceptable number of genes. Well, one measure, which is not the ultimate way, of course, recognize this, is not the ultimate way to get the information of whether we get all genes or not, okay, is to compare all the genes which are in your assembly, okay, with a database which is named BUSCO. Okay, there are also other databases. This is the most used, actually. There's also Sigma and other databases, okay, in which for every lineage, for example, for eukaryotes, for protists, for metazoan, for plants, bacteria, but also for insects, mammals, vertebrates, okay, for every lineage, okay, is specifically built, okay, a core set of genes. That is, we can expect in the 100% of instances in which we sequence, for example, an insect, okay, to find the 100% of those couple of thousands of genes that are in the BUSCO insects data. If we get less than that, okay, we can already say that this is a partial genome sequence. If we get the 100% of the genes in that data set recognized in our assembly, that we can say that, well, if the core data set is completely represented, it is very likely that we got, if not the 100%, 90–95% of the whole genes, based on the assumption that if we miss something, we miss it by chance. Therefore, both core and non-core genes, okay, have the same probability of being sequenced or missed in sequencing. Okay. Therefore, doesn't give you the complete certainty to get the 100% of the gene sequencing, but it is a good estimate of the work done. Okay, we will see after the break how it works more practically speaking. Okay, now, if you have any question for this part, we can talk about that now, or can we have a coffee today? Let's say 15 minutes, and then we'll start again.
Non vuoi cercare davvero?
Davvero? Lassù il tavolino?
Tengo un momento nel microfono.
Okay. Come on, you almost made it. So, Busco, we're talking about the method for assessing the completeness of a gene. Okay, as said, there are several methods. Okay, of course, using Busco only guarantee an estimates about what probably is more interesting for us, not necessarily the most important part of the genome, you know, okay, but for sure is what is important for us. Okay, just to retake from where we interrupted. What is Busco? Busco is a database. Okay, I say this is not the only database for task. Okay, it is just the most implemented and the most used for some lineages is the most complete. Busco is a database of so-called core set of genes. That is, these are for those who have confidence with genetics is probably the database of what were known as housekeeping genes. Okay. The housekeeping genes are those core genes, okay, that are important for all the cellular processes. Okay. Busco extend beyond the housekeeping genes by including all those genes that are always found in species, sequenced species, of course, okay, that belong to a certain lineages. Okay. It is clear that based on the level in the systematics of the tree of life we are working with, okay, we have a different number of Busco genes. Okay. Eukaryotes dataset, for example, is not particularly rich. It includes all genes that are always found in eukaryotes. Okay, there is a huge number of eukaryotes, you may know. So the number of genes in this set is not particularly large. It is not listed here, but for example, if you get the Busco genes for the hymenopterans, bees, wasps, ants, okay, you have more than 5,000 of genes within the Busco dataset. That means that it is a core set of genes common. It is shared by all hymenopteran species, or at least from all the hymenopteran species for which we have a genome sequence, okay, which amount to 5,000 genes. Imagine to sequence a new hymenopteran, like whatever ant species, you know, there are so many ants, okay, it is likely that if you select one, it's still to be okay. Therefore, you make a de novo sequencing, you assemble that genome, okay, and you compare the genes within your genomes, okay, with the Busco dataset for hymenopterans, okay. If you get the 100% of the Busco hymenopteran genes within your assembled genome, okay, then you can say that your genome is highly complete. That doesn't mean that you sequenced all the genes, of course. But it's a good indication, since, as we said before, if you include or miss a given genes, it could be just a matter of chance. Okay. Sequencing is blind, sequencing is random. Okay. If you miss some region of the genome, it is because that occurred by chance. Okay. So if you have 100 of Busco genes, you can be pretty confident you have, if not 100, say more than 90% of the genes sequenced. Okay. Comparing with Busco genes is simply more than that, actually. This is a classical view. Okay. Yes. Of course. Yeah.
But it's not running at all because it's stopped. So I'm wondering, have you ever tried Busco and it run without any problem, or if it is being isolated, for example, like that, it's or any problems like this?
Let's talk about that. I mean, first, which kind of database you use for comparing with this?
No, I don't remember exactly.
Okay, it will be a problem of the database. Okay, you can try looking within Busco datasets. Okay, if there is a list that.
Yes, this is for sure.
Okay,
We checked it because it's my run and try this analysis.
okay, but it's running out of memory or is.
Yeah, it says that it's running out of memory, but it's strange.
If you try to run Busco, it will be a technical problem, not related to the genome. If you try to run Busco genome on same time, okay, it will be a problem of installation of dependencies or the administration of. Otherwise, you can get an indication, not as precise as running Busco locally. Okay. If you look on Google for Genomics, is an online platform which runs a set of methods for assessing the completeness. Okay. If you go to the analyzing the analysis tab, okay, you can upload your genome there. Okay. You can select a set amount of Busco. In the case of Busco, you can select also any kind of sample that to be used. Okay. You launch the analysis, you put your email, often you have to wait for 1 day or 2, depending on the key on the server. Okay. And it gives you exactly the same analysis as the local method, with one difference which could be relevant. Okay. You know that running Busco output also the Busco genes found in gene. Okay. If you run it locally, you can use the meta-eoc flag that you that's meta-eoc, it's a flash meta-eoc, okay, give you the amino acid sequence of the gene and the nucleotide sequence. Without meta-eoc, you only get the amino acid sequence. And on Genomics, you can only get the amino acid sequence. That's the only point. But this could be helpful in case you have some problem on running locally, you can run it there. Maybe amino acids is enough.
Yeah, I have to ask you.
No, of course.
But yeah, the problem is that it runs out of memory, but it's strange because
Yeah.
so I was wondering if it was an old version or something like that.
I have to say that I don't know exactly the tips for the installation of the Busco scales. Okay, but maybe you have to open one of the install files to set the maximum memory allocated for that.
Try this.
Yeah, without the results.
Okay, we'll try that one.
So we're saying that running Busco, of course, it's not simply looking at how many genes you get from the, because there could be many different instances. The most common instances is that when you sequence a genome, not chromosome level, okay, it is possible that you get also fragmented genes. The end or both end of a scaffold could contain part of a gene. So when you run Busco, you have information about the complete Busco genes, okay, and the fragmented Busco genes. That is, of the gene of that data set for Hymenoptera, we are saying Hymenoptera, okay? If your genome is 100% complete, 100% accurate, chromosome level, and whatever it is, you should get 100% of complete genes. Okay? If it is not that the case, it is possible you have a percentage of fragmented genes. Most common situation you can find in literature, for example, is to have Busco genes about complete genes 90%, then you have 3% of fragmented genes. That's quite common. That doesn't mean that you get a shitty genome, but it is still a good genome. Okay, a little bit more complicated when you have a number of duplicated genes. What we can expect to be a single copy gene, okay, it is a single copy gene until we found a genome with a duplicate gene. Okay, gene duplication is much more pervasive than we might expect. Okay, therefore it is possible that some of the single copy genes or well-known copy genes, okay, exactly in your genome are duplicated, okay? And, well, this is not necessarily a bad news, but still it is not a good news, okay? Because duplication, if it is true that is a common biological process, could be also a problem of assembly artifacts. That is, something during the assembly goes wrong, producing a gene duplication which is not actually true. Okay, it is duplicated in your assembly, but in the real genome is not duplicated. Okay, therefore, are those situations that when you have a significant fraction of Busco genes that are duplicated, it is better to investigate more in detail the story. Okay, there are methods that can be used for assessing whether there are artifacts. Okay, of course, they are long methods and time methods. Okay, difficult to analyze, but there are still methods. Okay, and then the statistics we always hope for being zero. Okay, is the missing Busco genes. It's legit. Okay. If you have 5,000 genes in the Busco dataset, okay, our best hope is to doesn't have any missing gene, of course. Okay. Nonetheless, it is possible to have some gene missing. That could occur for two reasons, basically. Most common reason is that we miss the sequence portion, even a small portion of the genome. Sorry. Okay. Therefore, we miss some of them. Okay. If we have a 1% of missing Busco genes, let's make a big assumption, we can say that we lose the 1% of all genes in the genome. Always assuming that missing some genes is a matter of randomness, of course. Okay. There is also another point to consider here. Okay. Busco genes are those genes that are found in all sequences of Hymenoptera, okay, in our example. Okay, if we add a new Hymenopteran, okay, it is possible that the database should be reduced by 1 or 2 genes. Okay, it is not say that if we miss some genes, automatically we miss some part of the genome. Okay. It is possible that exactly those genes are missing in that species. And if we sequence more species closely related to that one, they also miss exactly those genes. So the Busco dataset, like the Sigma dataset, like many other pretending to be complete and absolute dataset of genes, okay, actually are dynamic dataset. They change as more genes are sequenced and analyzed. Okay. If you get a chromosome level genome with a number of scaffolds, which nicely approximate the number of chromosomes. And if you miss some Busco genes, it is very likely that that missing Busco genes really are missing from that gene. Okay, so the point is, if we miss a few genes, okay, depending on how many genes we are missing, okay, but it is expected. It could be expected, okay, to miss something. Therefore, it's not a tragedy if on 5,000 of genes we miss 3 or 4 genes, okay, that lead to less than 1% or 1% of Busco genes missing. That's okay. Maybe it's not that important, but I mean, shouldn't be a blocking warning with this one. Okay, that is a classical scheme in which we used to depict, okay, the Busco results. Okay, the longest bar of complete genes, okay, the longer the bar of complete genes, the better the sequencing. Okay, even if we can expect some duplicated genes and some missing genes, okay, if we usually have the complete statistics approaching the 100%, let's say more than 90%, okay, well, that's a very good job. Okay, we can consider our genomes complete. Okay, and everything in between, of course. To get an example, okay, RNA sequencing. It's possible to make a Busco analysis also on the transcriptome. You get the transcriptome, you get the genes from the transcriptome, okay, and then compare with the Busco genes. Okay, if your RNA-seq experiment include, for example, the whole organism or several tissues of the organism, you should get the sequence of all the genes in the genome. Okay, therefore, here it is expected to have a huge quantity of complete genes. It is more problematic to have missing genes in an RNA-seq experiment than into a genome experiment. And usually, if the job is done properly, RNA-seq approximates the 100% of complete genes. Of course, also in this case, it is always a matter of a trade-off in getting a good job and doing that 10 times in order to have all genes. Okay. Summarizing about the assembly quality, okay, this is the 3 points that you should always ask yourself after the assembly. Okay, how much the genome is sequencing? It's a very difficult answer to give this one. Okay, for all those reasons we talked about right now. Of course, if you have flow cytometry or fluid coloration, okay, experiments telling you that the genome of your organism is, let's say, 1 billion base pair, the so-called gene size, and your genome assembly is 999 million something base pair long, okay, which means that you sequence 99% of the genome, okay, something is missing. Probably the missing part is negligible. Okay, then you know. The Murphy law say that if you miss something, that missing something is the most important thing in your experiment, okay? But that's not the story, okay? Therefore, how accurate is the sequencing? Which is often how covered is the genome, the so-called genome coverage. Well, genome coverage is a sort of term which has two meanings. The genome coverage could be how much of the genome has been sequenced, okay? But usually is intended like how covered. That is what we said before, okay? Every single position, how many times has been sequenced. It is how many reads cover that specific nucleotide position. The higher the number, of course, the better, okay? We can calculate the expected coverage based on the output of the raw data and the genome size. There are some maths to do, okay, that can give you an expected coverage, okay? But usually while assembling, okay, the same assembler algorithm output also the coverage of every single scaffold, okay, which you can average across the all scaffolds, okay. How good is a coverage? It depends. Let's say that an acceptable coverage for a genome is for sure higher than the 50-fold coverage. But we know that sometimes it is better to be more, say, gentle. It is, if we cannot reach the 50, but we have the 40, we just say the coverage is 40, that is the result. Okay. Then someone will say it's okay, or it's not okay, or so on. So, contiguity, it is how many scaffolds we get, okay, and how long they are. The lower the number of scaffolds, the higher the N50 statistics are, okay, the better. Just remember, if you know, it is not always so, but if you know the chromosome number, approaching the chromosome number means that you are doing a very good job, okay? In the most of the case, all the genomes already available in the databases, okay, like in CBI or in a database, okay, doesn't even approach the chromosome number. Genome assembled only with, say, long reads, like the termites we saw before. Here, you see. Here are 427 scaffolds for a species which harbor 36 chromosomes. Okay. If we add an IC sequencing to this assembly, it is very likely that this number will drop below 1. Okay, which is a better approximation of the chromosome number, of course. Okay. Then we can refine further and further. If we have, let's say, 80 scaffolds and 36 chromosomes, it is possible that one of these scaffolds is the mitochondrial DNA, which can put apart. Okay. Some of the scaffolds could be symbionts, which we can put apart. Okay. Some of the scaffolds are a second sequencing of a chromosome. Let's say that we have phased chromosomes, therefore we have 2 sister chromosomes. Okay, now we can put it apart. Okay, and we can reduce, sometimes manually, sometimes computationally, okay, approaching the chromosome number. That's the meaning of genome contiguity. Okay, and then once we get our genome assembled, okay, we start to go downhill. Okay, assembling the genome is the hardest task. Okay, usually, okay, in producing and analyzing the genome. Okay, after that, okay, it is everything a little bit more easy, like genome annotation. What is a genome annotation? Genome annotation is finding genes within the genome. When you get your genome assembled, you just get a collection of DNA sequences, scaffolds. Now, forget the number, okay? You get some bunch of scaffolds, okay? Well, these scaffolds harbor genes. The same genes that Busco tell you, you have 90% of genes, okay? Therefore, in those scaffolds, if you don't have 100% of the genes of that species, still you have the 90% of the genes of that species. You know how many genes there is in an animal genome? It is between 12,000 and 30,000 genes, depending on the species, of course. The human genome is about 16,000 genes, I think, or something like that. Okay, and you have to find that. Okay, this is called annotation. Annotation is a task which includes the use of one or multiple tools, depending on which kind of species you are dealing with, what you are looking for. Okay, there are not only genes to be annotated, but there are also repeats to be annotated. Repeats, like transposable elements, also harbor genes, but those genes are not genes of the organism, are the genes of the transposable element. Okay, and we have to get rid to that. Okay, therefore, typically, an annotation run, okay, start with annotating the transposable element, which is done in different methods, often with comparing with known transposable elements. Okay, then we say we mask the genome of transposable elements. That is, we deplete genome with the region containing the transposable element, and then we run the annotation. Running the annotation means finding genes. Finding genes is not like finding genes in the transcriptome. In the transcriptome, you get the RNA sequence, so you have to find the operating frame. Every operating frame is a gene. Often, it could be also of a splicing variant, okay? But still is treated as a single gene, right? Finding the genome, the genes within a genome is a little bit different, because within a genome, there is not only operating frames, there are also introns and UTRs. That is the untranslated region, which are downstream and upstream the gene. Okay, the genome is much more. The gene is much more than the coding sequence. Okay, and this can be done through different methods, which I not list here, but know that there are different methods. Okay, starting from the simple comparison with known genes to structural search of particular genomic architecture which underlie the presence of a gene. Okay, which are listed here. But most important part and interesting part of the genome annotation, okay, is the functional annotation. Knowing that in that region there is a gene, okay, it could be interesting, okay, but if you do know what's the role of that gene in the cellular process or in the organismal processes, it's not particularly useful. Okay, so there are two ways to assess the function of every given gene. The most fascinating way is to perform functional experiments. Okay, which means expression experiments, which means knockdown experiments, etc., etc. Okay, if you multiply these experiments for, let's say, an average of 16,000 of genes, okay, a couple of scientific life, okay, could be perhaps enough. Okay, if you don't have so much time, okay, then you can run comparisons once again with database. Okay, database which harbor information about the sequence, the structure, and the distribution of known genes of known function, typically model organism, Homo sapiens, Arctus norvegicus, Saccharomyces cerevisiae, Drosophila, C. elegans, etc. Okay, where functional experiments have been carried out, okay, therefore it is known the function, okay. This is used for transferring the functional information from that gene, okay, to the gene of our genome, based on the fact that similarity underlie similarity in function, which is true? No, but it is an assumption, and therefore it is legitimate to use it. Okay, that is the core also of the orthology conjecture, which we already talked about, and we will see in a few moments. Assigning, for example, gene ontology terms, so-called geo terms, okay, means to assign a function to our genes. Okay, and therefore we can group our genes based on their functional association. A gene ontology term could be a function linked to a biological process, to a molecular process, okay, to a cellular process, depending on, yes.
Is there any percentage of error when assigning a function?
Yes, very large, actually.
Okay. Is it known according to the use that they tell you?
Yeah.
Okay.
Let's say that assigning functional terms to gene is a random functional notation is a minefield. Okay. Everything is true, everything is false, because it's a purely statistical model. Okay. And many functional terms are nested within other functions. Okay. So depending on the level where you stop your analysis, which is arbitrary. It's a part of this gray zone between purely scientific method and observational. Okay. The level in which you can make a correlation, okay, is a level you can consider. The only important thing is that you disclose the way you select the functional annotation. Okay, that's it. We all people know that GO terms are not defined with the ultimate functional annotation. Okay. But if you find an association between a gene which has a GO term which is linked to the aggressiveness, for example, okay, and it is a gene in well-known organism with an aggressive behavior, and this gene is more represented in the genome because contain many duplication, okay, that is not a definitive evidence, okay, but it is a suggestion, okay, of something which could be true, of course. Okay. In these instances, my suggestion is always to make hypotheses, propose models, okay, without using a tombstone on the story. Okay, it is not that I found the gene of aggressiveness in humans. Okay, because in best of the case, even if it's everything's true, that is a gene which is linked to a cellular process which activate during the moment in which that human exhibit an aggressive behavior, which is a couple of million years of evolutionary history in all the story here. Okay, therefore it should be careful in make bold statement in this, but it's logical to get honest indication. Okay, if you find a correlation, there are also statistical methods that tell you which is the level of error associated with that correlation. Okay, if it is an acceptable error, it is legit to make that assumption. That's all. Well, this is what we were talking about here. Okay, that's for give you an idea of what are, all the possible role and function
of geoterm that could be assigned to genes within a genome. And well, I would like to keep better to some part. You can see this by your own level. You will have the slides. That is not very important thing since time is running out. I will run, for example, here, okay, directly on comparative genomics, okay? And would like to show you, okay, something that we talked about. First, the genome analysis, okay? We talk about difficult gene to assemble, we talk about the use of some assembly, some sequencing strategies that take condition, or it is better to use another strategy in different conditions, okay? This may depend on what is your biological question, of course, what is your aim, is assembly of a genome, or simply remapping on a reference genome for getting population not data. This is also good, right? Discriminate which kind of sequencing. Sometimes knowing, for example, the nature of the genome you are analyzing, okay, you can take it as a suggestion to which kind of sequence to adopt, okay? And this is strictly linked to the kind of genome you analyze. What you can find in a genome, okay? In a genome, you can find for sure genes. On 3 billion base pair human genome, you have 16,000 genes, which means that less than 4% of the human genome is made by genes. Okay, so what is inside the remaining of the genome? In the remaining of the genome, there are a lot of stuff we are not potentially interested in, okay, like just possible elements, like centromeric repeats, and these part of the cake are gene bursers. Many genomes have very large gene bursers. It is portion of chromosomes where you cannot find any trace of a given gene. There are no operating frame, there are no repeats, there is nothing, it's just run. That's part of the so-called junk DNA. There are not any promoter or enhancer. Promoter or enhancer are part of genes. Okay, so there are not always regulatory agents here. Okay. These are really gene bursers. Okay. And so you see and this is just human gene. Okay. You have to see the biology. Genome is a help. There's plenty of things that are genes intermingled with the transposable element which contain tandem repeats dispersed within genes. It's a really a nightmare to assemble a mollusk gene. Okay. That is when I say it's a difficult genome to assemble. Well, mollusks probably are one of the most difficult genomes to be assembled. Okay, we start to get proper assembly of mollusk genomes with the advent of long reads. With long reads, okay, everything is better. Okay, but before the long reads, to assemble the first oyster genome, which is not particularly big because it's less than 1 gigabase, okay, this is less than 1 million of base pair, okay, has been assembled with only short reads, okay. It took several years and an amount of sequences which is much more than expected, okay, because the genome is completely disorganized, let's say. Okay. Therefore, within a genome, there is a lot of stuff, okay? And then there is the mitochondrial DNA. The mitochondrial DNA is actually the simplest part of the cellular gene, okay? It's very small, especially in the metazone. In plants, it's slightly bigger, okay? But it is very easy to get information about mitochondrial genomes, and sometimes it is enough. If we want to analyze, for example, the phylogenetic pattern of a given species complex or given group of species, often getting the mitochondrial genome is enough. It is likely that a very random, low-pass, short-reads sequencing, okay, it is enough for getting the whole mitochondrial genome covered more than 50-fold. Okay, in metazone, the mitochondrial genome is between 15,000 and 20,000 base pair, depending on the species. On average, it's a 16, maybe 17 base pair. Okay. The number of genes is basically fixed, especially the protein coding genes. Okay, there are 13 protein coding genes. Okay, there are always those 15 protein coding genes. We know only few instances in metazone, at least. Okay, in which there is 1 or 2 genes missing. To my knowledge, there are no more than 13 protein coding genes in the mitochondrial gene. Okay, if you want to get the mitochondrial genome, you can analyze short reads. An example is better than memory. Okay, sequencing on of the chimpanzee. Okay, including the millions of short reads, millions. Okay, and if you isolate 1 million randomly of short reads from the chimp genome, okay, it is enough to get the whole mitochondrial genome covered more than 34. Okay, so it's very easy to get it. Okay. So why to use mitochondrial DNA or nuclear DNA? This is a story you probably know better than me. That's a very, very old story, okay? Mitochondrial DNA is much more variable, therefore it is possible to use it for analyzing closely related species. There is no recombination, which sometimes is better. For example, in the phylogenetic framework, getting rid of recombination is better because recombination leads to a reticulate pattern that completely bias the analysis. It is applied, so you don't have several leaves, you just have a single allele in principle. The tissue is very complex. Okay, there are no introns, there are no regulatory regions between genes, there are no transposons, there are no repeats. Okay, there are only genes. Obvious orthology. This will better explain later. Obvious orthology means that 13 protein coding genes in the mitochondrial genome of animals. Okay, well, these are always those 13 protein coding genes. So there is no matter to finding orthologs. You know they are orthologs. The problem is that is half of the story. Because mitochondrial DNA only tells you the story of females in animals. Great. So you know the evolutionary history of females, assuming that the evolutionary history also of males, okay, is overlap of those of females, which is true in many cases, not always. Okay, nuclear DNA is surely less valuable. Okay, it evolved slowly with respect to mitochondrial DNA. There is a lot of recombination. I think it is deployed, sometimes more than deployed, because there are triploids, hexaploids, animals, whatever, and it is a mess. Repeats, possible elements, gene desert, and whatever it is. But information stored within nuclear genome is much more higher than the one you can find in mitochondrial genome. And if you are looking for different species, okay, what makes a species different? The genes into the mitochondrial DNA or the genes within the nuclear genome? Of course, the second. Genes are different not because they have divergent mitochondrial genes, but because they have different genes within the genome. Okay, therefore, if you want to have large information, the nuclear gene is better. How many genes there are in a given gene? Say, say later that usually metazoan species, okay, we can found between 12 to 1300,000 of genes and 12 to 30,000 genes. So it's like okay. Well, this number can vary. Different species have different genes. These are not missing because of technical problems. Okay. These are missing because they are missing. Okay. If you have two sister species, one having 15,000 genes and the other having 900 genes, it is not that in the second species you miss some genes. You have all genes. The first species have more genes. Why? Because genes pop up and regenerate very quickly sometimes. Sometimes they make the difference between one gene and one species and another. And the number of genes are not always correlated with the genome size. Larger genomes don't necessarily harbor more genes. Okay. Larger genomes may be larger for other reasons. An example, Aedes albopictus, the tiger mosquito. The tiger mosquito, you may have population having genome size of 1.5 billion nucleotides, and other populations with genome size with more than 2 billion of nucleotides. The number of genes is exactly the same. What's the matter? The matter is that the number of transposable elements increase so much in few generations to produce this disparity in genome size in population of the same species. Okay, that's tell us that the genome size is not proportional or not necessarily proportional to the number of genes. Finding orthologs. This is one point which is very, very important. Okay, once you get all genes from several genomes and you want to make some comparative genomics, okay, which genes you are going to compare with which genes of the other species? That's not an obvious question. We already mentioned the obvious orthology in the mitochondrial DNA. Mitochondrial DNA in animals, at least, always, or the best, most of the case, 99% of the case, have 13 protein coding genes, and these 13 protein coding genes are always those one. The ATP6, the ATP9, 8, and there is the cytochrome oxidase, subunit 1, 2, and 3. Cytochrome oxidase B, and NADH 1, 2, 3, 4, 5, 6, and 4L. That's all. Okay. The cytochrome oxidase subunit 1 of one species is for sure ortholog of the cytochrome oxidase subunit 1 of another species. Cytochrome oxidase subunit 1 of Homo sapiens is for sure ortholog with the cytochrome oxidase subunit 1 of Drosophila melanogaster. There is no doubt about that. And you know why? Because there is no gene duplication in the mitochondrial DNA. Every gene is always single copy, which is not true for the nuclear genes, where the gene duplication is really perfect. So when you compare genes across different genomes, you have to know that A given gene within a genome may have 2 copies in another genome or 3 copies in another genome. Okay. And it is relevant to compare one gene of the species A with the copy 1 of the species B or the copy 2 of the species B. It is not the same. So you have to find which is the real, we say, ortholog of that gene. Okay, that's the meaning of finding orthologs. It is finding the correct counterpart of a given gene in the other species. A long story short, if you have a gene duplication here, okay, the gray area is the evolution of the species. Okay, the colored lines is the evolutionary pattern of genes. So here you have in this species, a single species ancestral to human and the chimp. Okay, you have a gene duplication. Histone 1 H1.1 H1.2. Okay, so from this moment on, you have 2 genes within this lineage. Then you have the species split. Okay, and you have 2 copies in the human and 2 copies in the chimpanzee. Okay, that's the best of the instances of gene duplication you can find. Okay, so the copy red in human is ortholog to the copy red of the chimp, but it is paralog to the copy blue of the chimp. And the copy red and blue in the 2 genomes are paralogs. These are in paralogs and out paralogs here. Okay. So it is relevant. It is important to know that we have to compare the red gene with the red gene of the chimp and the blue gene of the human and the blue gene of the chimp, and not discuss between the 2. Otherwise, we are comparing something different. Because during the evolutionary history of these 2 genes, blue and red, okay, they may experience different evolutionary gyra, leading to different evolutionary patterns. Okay. Therefore, if you find some divergence between paralogs, not necessarily reflect the divergence between species. Okay. The only comparison between genes reflecting the divergence between species is the comparison between orthologs. Orthologs are homologous genes originated by speciation. Paralogs are homologous genes, but originating by duplication. Okay. That could seem silly to talk about that, okay, but it is the foundation of comparative genomics. It is so important that we are still working on the concept of orthology. Okay. At the moment, we base the principle of orthology using the so-called orthology conjecture. While we know perfectly what does it mean orthology, okay, no one of us was there when the human and the chimp split. So no one of us can tell which is orthologous to which. Okay. Because orthologous originate by speciation. Okay. We have to find a way to infer orthology from the actual data. Okay. The orthology conjecture tries to explain exactly this point. Okay. The orthology conjecture is, as they say, a conjecture. Okay. That is, we assume that orthologous genes retain high similarity and the same function. Okay. Therefore, there are methods, sometimes very simple, sometimes quite complicated, okay, that can compare all the genes of one species against all the genes of the other species, okay, and output a set of genes of species 1 and species 2 that are orthologs based on the sequence similarity. If they are similar enough, they are considered orthologs, okay, and thanks to the ontology conjecture, okay, we assume they share the same function, which is the principle through which we pass functional notation from the Drosophila genes to the whatever genes. Okay. Once we get all orthologs, we can do whatever we want about evolution and the assessment of the gene component of the gene. Starting from the gene stratigraphy, okay. The gene stratigraphy is a kind of analysis, okay, which can tell us, for example, okay, which genes in the given genome are shared with other species of the same genes, other species of the same family, other species of the same class in the family. Okay. These are sequencing of crustacean. Okay, this is another crustacean. Okay, you can see here these are genes that are shared by these two species. This is the number of genes that are shared within these two species. These are the number of genes that are shared between those species here. Okay, you will say, and what? Well, these genes here probably are the genes that made these species different from this one. Okay, these in this data set, okay, are the genes that characterize only these two species. Okay, and therefore more likely are those involved in the biological processes that make these two species similar to each other but different from this one. Okay, that's the principle of stratigraphic analysis. Okay, this is another way to see. Okay, these are genes, okay, that are present in many mammals but not in humans. Means that humans at a certain point of their evolutionary history lose those genes. Okay, these are all those genes that are shared among placental mammals, okay, that are missing, therefore, okay, in this critter here. These represent evolutionary novelties. Okay, that is, genes that are present also only in primates or only in carnivora. Okay, so analyzing these genes, okay, you can find something about the evolution of the specific lineage. Another interesting point is to analyze gene families evolution. Genes are, now we know that the single copy genes are actually the lesser part of the genes in a genome. Most of the genes in a given genome belong to a gene family. A gene family is a set of genes all originated by the gene of a duplication of an original genes. Globulin genes, for example. Okay. These are gene family. Analyzing gene families expansion and contraction, that is how many duplication occur during the evolution, okay, can tell you something about the biology of those species by making functional notation of expanded or reduced gene families. Okay, a given gene family, for example, okay, could be lowly represented on this branch, okay, and become more represented on this one. It means that during the evolution from this point to this, okay, there was a contraction and an expansion of the gene family, probably linked, okay, to some specific function of that lineage. Okay, if you run a functional notation, you may discover that that gene family may be linked to a specific process present on this species. Here, for example, termites, again, okay, what it is important for termites? Termites are social insects, okay? There are two things in termites that are very important, okay? Communication, because within the colony, they have to communicate with the other colony members, okay? And termites live in wood and eat wood, okay? Therefore, they have to do with all the defensive compound present in the wood, okay? So there are two gene families that are very important for termites, and are odorant receptors, because they mediate the communication, and P450 genes that are genes all linked to digestion of drugs, xenobiotics, chemical compound, whatever it is. Okay, and here you can see, but well, it's not easy to see that there is a specific amplification of these gene families in termites. Okay, according to the fact that the life history traits require more odorant receptor genes, more P450 genes. And this is another example. You can recognize it here, the Bushko assessment. Okay, here well, there is more numbers. There is expansion, contraction, and this is a depiction, okay, on gene functional mutation. And you can see that many of the expanded gene family is a defensive response against bacteria. Okay, and if you have a look on this paper, you may realize that these species live in an environment which is plenty of gram-positive bacteria. Okay, or it is an adaptation. And now the last part, okay, which is more specific for finding local adaptation, okay, is of course the population genomics. Doing comparative genomics is not simply analyzing genome composition, different genome architecture, evolution of gene families, expansion, contraction, whatever, which genes are present or not in other species, okay, but it is simply also finding polymorphism, which may differentiate different populations, for example. Okay, that could be important. It is the primary importance in forensic science, of course. Finding polymorphism at the genome level help to identify biological traces on the crime scene, for example. But less dramatic, okay, is the use of population genomics for analyzing fishing stocks. By analyzing population divergence in different fishing area, you can realize that some stock of fish come from a given area and not from another one. Or you may realize that those species are not actually the species you are selling, but it is the sister species, okay, which from rules way to approach the problem could be important. Last thing, okay, with population genomics, you can find, for example, divergence across population, lineages, species, okay, and you can find the instances of gene flow between population or, more important, hybridization. That's the old story because of the hybridization between wolves and wild dogs, for example, okay, which may be problematic for conservation issue, for example. Okay, so analyzing the genome with a population genomic approach, okay, you can realize that, for example, in that part of which I don't know where these have been analyzed, but there is no hybridization with the dog, okay? If not in this population where you can find the red component that belong to the dog. Okay, that means that once or a couple of times in the past, okay, was an integration of dog alleles into the wolf genome, meaning that somewhere, okay, dogs and wolves hybridized. Okay, and now fish. Let's look at that. What to do with it? Tracking the food. Okay. Not a big thing. Okay. Couple of fish. Not very long. Okay. Not necessary. What to put
while writing a small project in comparative genomics. The biological question, why I'm spending so much time and money for getting sequencing, for getting novel assembly, what I want to analyze. Would I analyze the divergence between population? There is a suspect integration of replicating gene with everything human gene. Okay, so try to imagine, maybe based on your experience or based on your research topic, okay, if there is something that could be addressed with a comparative genomic approach. Okay, the biological system. I like crustacean. Okay. Let's find something interesting in crustacean. Okay, that would be together with biological question. Okay, crustacean lineage that have been never sequenced so far, which may be important for let us understand better the complex, just the evolutionary history of crustacean. That would be very interesting. Okay, and the sampling strategy. It's very, very important. You can never get back from a bad sampling strategy. Okay, for the sampling strategy, have to be done very, very, very well and accurate. Where to get the sample, which sample to analyze, and look, sometimes is relevant to select a male or a female for sequence. Because at the chromosome level, male and female could be different. You know that in Homo sapiens we have a heterogenetic sex, therefore we have different chromosomes. In other organisms, males have one chromosome less because it's 0. That's determination. In some species, like ants, for example, males are haploid. So if you have to choose to sequence an haploid or an diploid organism, which organism will you select? The haploid, because you have only one genetic variant to sequence. More easy, okay? If you don't have haploid genomes, you always have diploid genomes. Okay. What to do? Isofemale lineages. Can I bring in lab my organism to get a fully homogeneous genome, which is exactly the same story as having a haploid? Yes, I will do that. No. Okay. And approach the thing. Anyway, the kind of genomic approach doing the novel assembly require sequencing which is different from resequencing, knowing that there is already a reference genome. How do I know if there is a reference gene for my species? Only from the CBI or in a database. And then you will find whether there is already an assembly which can be used as a reference. Sometimes it's also labeled as a reference. Okay. 4. My genomic approach is a total approach. I first plan to make a de novo assembly for the reference genome, and then spend more time and money for the sequencing and mapping for doing population genomics. Okay. Of course, you can do whatever you want. Is an essay. Okay. And then I would like you to list the expected results. That is, why should I approach my biological question with these tools? Why should I address my scientific question using a genomic approach? Because I expect to have certain results, not because it is cool, not because
everybody now sequence genomes, and therefore I will sequence genome for doing the same thing. But I use a genome approach because I expect to have this result that without a genomic approach, it is not possible to get. Okay. This said, if you have any doubts, if you have any question, you can write me, okay? We can discuss it by email or by conference call, maybe, okay? To try to go deeper into the point. Okay? Feel free to contact me anytime. And thank you for your attention. We have like 1 minute for questions. Yes.
Yes.
I actually thought it's going to be out because
No, but there are some patterns we may recognize. To report increase. First, the kind of, for example, time the kids usually tends to accumulate close to structural part of the process. Okay. In few instances, we can find also probably precision. That is small cluster deviations. Not very long. Okay. Anyway, they don't really very large. Very large. Very large in class. Just possible evidence usually distributed by the element across the course. When transposed. But after a few generations, most of them, okay, are eliminated by population, by population, or one population by population of molecular process. Okay. So you can actually observe their distribution more than certain time. Okay. That, of course, may have an impact on the chromosomal life history, because clusters of repeat usually are of auto-accumulation. Therefore, you may expect that at the level of a positive cluster, you not only have an increase in the combination rate, but also an equal rate. Because being recruited, okay, may misalign chromosome during the phase. Risks that will lead to longer recovery. In the most dramatic cases, this will include also the ablation of genes or duplication. Then it is the chromosome morphology. The way these genes distribute after from chromosomes may be different from the classical chromosomes in sampling. So, saying that there is a pattern or a rule that holds for all instances, it's very good. Okay. But for sure, there are some. For example, in mosquitoes, okay, we observed this cluster of Champagne Neurosophica, which is a sister lineage. Therefore, it also is a feature of this. And to my knowledge, the same cluster of possible elements are not actually It's here, okay? And so you're going to start.
La registrazione.